So a very good morning to Dr. So today I am going to talk about the GWAS of severe COVID-19 with respiratory failure. So now I will introduce the virus that can cause the COVID-19. So the virus was named by the World Health Organization, which called the SARS-CoV-2, and the disease was named by COVID-19. So this COVID-19 outbreak was start at the Wuhan, at the China, and the country China. So now the virus has spreading to the whole world and infected many people. So with this, when we infected by this virus, so we have the 14 days incubation period. So during this period, the virus can spread from one to one another. So besides that, the symptoms usually are like fever, coughing, and the severe one, you have the difficulty in the breathing. So in some patients with very severe condition, they will need the mechanical ventilation in the ICU of any machine that help them to breathe. So we have found that some people, when infected with this virus, they will develop mild symptom, but others will develop very severe symptoms such as the respiratory failure. So in order to know the genetic factor that associated with this SARS-CoV-2 COVID-19 disease, so I am going to propose a GWAS of severe COVID-19 or respiratory failure. So first of all, I would like to say that every people have the genetic variation. Like example for the SNPs are a gene factor that contribute to genetic variation among the individuals. So in this GWAS, we will determine which of the SNPs are more associated with more risk to this severe COVID-19 of respiratory failure. So before carry out the GWAS study, so there are some key ethical issues that need to be taken into consideration. So such as the informed consent, the feedback of the result, and the privacy of the participant. So for the informed consent, the participant will be informed about the whole process and the whole method that will be carried out during the research study, and they should know all the details that will carry out during the research study. So for the feedback of the result, the participant have the right to know the result after the research study was done. So the researcher can have a result discussion with them after the research study was done. So and the last part is the privacy of the participant. So the privacy of the participant will not be released by the researcher in their published paper, so that this will protect the privacy of the participant to prevent the leak of the information of them into the public. So in order to carry out the GWAS of the severe COVID-19 with respiratory failure, so we will recruited about 1,000 healthy people in the Malaysia and 1,000 of infected people in the Malaysia. So the healthy people will act as the control, the infected people will act as the case. So this sample collection was based on different age group, different blood group, and different ethnicity. So after the people were recruited finished, so all the individuals have to carry out the nasopharyngeal swab to determine the status, whether that they are infected or they are healthy, to confirm their status. So the confirmation test was the real-time reverse transcriptase PCR. So this is to detect the presence of the SARS-CoV-2 virus in their body. So after the confirmation of the status of the individual, so the blood of the individual will be collected to do the DNA extraction process. So during the DNA extraction process, we are going to use the automated magnetic bead method by using the Perkin-Elmer Chemagic MSM1 instrument. So with this instrument, it has a valve designed to prevent the cross-contamination of the sample. Besides that, it is also time saver because it can run up to 96 samples per run. So after the DNA extraction process was done, so we are moving into the whole genome sequencing. So by carrying out this whole genome sequencing, we will identify the genome of each particular individual. So this whole genome sequencing method, we are using the next generation sequencing method because it is time saver, because it uses the shorter time to develop large amount of the data to support the assembly of the genome. So after the whole genome sequencing so we can identify the SNPs in each individual. So after this, we are doing the imputation process. So by doing this imputation process, we need to ensure that all the SNPs are not missed in the process, so that will not the accuracy of the result. So after the imputation process was finished, so we will move into the quality control. So the purpose of carrying out this quality control is to ensure that the result of the GWAS was pretty accurate after this. So after all the things was done, so we will get the result. So the result we will can in the form of the quantum plot and the Manhattan plot. So based on the recent study, the study has stated that the chromosome 3 and chromosome 9 was playing an important role in the development of this severe COVID-19 with respiratory failure. So besides that, recently the statistic also show that the blood group A people will tend to have more high risk to develop the severe COVID-19 with the respiratory failure, while the blood group O people will tend to have a protective effect from this symptom. So as a conclusion, GWAS was an useful and important tool that help people know that which genetic factor that were associated with a particular disease. Besides that, GWAS was also an important tool to help people understanding the relationship between the genetic factor and a particular disease. So that's all for my video presentation. Thank you.